But what does the bacteria even look like? Bacteria have no nucleus. They are prokaryotic. They lack a nucleus. They still have genetic material. It's just not housed inside of a nucleus. It's housed inside something called the nucleoid region, which is just a region. These cells also don't have any organelles that are encased in a membrane. They may have organelles or little things inside of the cell that have special functions, but any organelles they have are not bound in a membrane. They have no membrane bound organelles. For the most part, the chromosomes that carry the genetic material for bacteria to survive is circular. And there also may be other material that is known as a plasmid that isn't necessary for their survival, aka for them to make energy or to reproduce, but may allow for them to live longer by evading antibiotics. All bacteria have a cell membrane, and outside of that cell membrane, they may have something called a peptidoglycan layer. If bacteria do have a peptidoglycan layer outside of their cell membrane, when stained with a Gram stain, they stain purple. So these cells that contain the peptidoglycan layer outside of the cell membrane are called Gram-positive. Cells that do not have the peptidoglycan layer outside of their cell membrane, they will not stain purple on a Gram stain. They will instead stain a pinkish red. These will be called Gram-negative. There's 3 ways in which a bacterium can change its genetic material from how it started. Bacterial conjugation, which is where one bacterium that has a plasmid can give a plasmid to another bacterium through something called the pilus. Transformation, which is where genetic material that's just freely floating in the environment, whether that's DNA or RNA, is taken up by the bacteria. Bacteria can also have mutations occur, but all of their polymerases have 3' to 5' exonuclease activity, which means they have a much better ability to proofread and prevent mutations from making it out of the DNA replication phase. Some bacteria evolved before the atmosphere had enough oxygen to create energy through cellular respiration. Any bacteria that does not require the use of oxygen in order to create energy is called an anaerobic, an meaning not, aerobe meaning oxygen, so not oxygen, an anaerobic bacteria. Any bacteria that does use oxygen in order to create energy is called an aerobic bacteria. Some bacteria have the capacity to choose whether or not they're going to use oxygen or not use oxygen, depending on the environmental conditions. These are called facultative. They're called facultative anaerobes or facultative aerobes. It means the same thing. It means they can use oxygen to make energy, but they can also not use oxygen to create energy. If the bacteria only has one choice, if it can only make energy through using oxygen, it is an obligate aerobe. If it can only make energy by not using oxygen, it is an obligate anaerobe. Obligate aerobes only use oxygen to make energy. Facultative anaerobes/aerobes can use oxygen, but don't have to create energy. If these bacteria are going to make energy, it has to come from some kind of a food source. If the bacteria can make its own food source, it is called an autotroph, self-feeding. Auto meaning self, troph meaning food. If the bacteria has to get its food from something else, it is called a heterotroph. Hetero meaning other, troph meaning food. Autotroph can make its own food. Heterotroph needs something else to make its food for it. If we assume an environment where there's unlimited amounts of food, unlimited amounts of oxygen, and everything else that a bacteria needs in order for it to grow, that bacteria is going to grow exponentially, where it's just gonna shoot up. However, once there's a lot of bacteria around, that bacteria is gonna start getting crowded. It's not gonna have easy access to all of its resources, and at that point, it's just gonna plateau. Unless we replenish all of those resources for the bacteria to grow again, eventually all of those bacteria are going to run out of food. They're going to run out of all of the other resources they need in order to live, and that population is just going to tank off. So bacterial growth curves have a phase where there's nothing happening, a exponential phase where a lot of growth is occurring, a plateau phase when they've reached like their peak point, and then a steep drop off once all of the resources have been used up. In summary, bacteria are a type of prokaryote, therefore they have no true nucleus. They also have no membrane-bound organelles. They are able to change their DNA through 3 processes: conjugation, transduction, and transformation. If they possess a peptidoglycan layer outside of their cell membrane, they are able to stain purple under Gram staining. Having a peptidoglycan layer means a bacteria is Gram positive. Not having a peptidoglycan layer means the bacteria is Gram negative. Bacteria can create energy with the use of oxygen, aerobic, or without the use of oxygen, anaerobic, and some bacteria have a choice, facultative. They don't have a choice, obligate. If a bacteria has to get its food from something else, it is a heterotroph. If the bacteria can get its food from itself, it is an aututroph.